All right, here is going to be an overview of aerobic respiration that you should be able to recreate on your own. We will be starting out with glucose as our starting molecule. That is the easiest molecule to harvest. And that glucose is going to be broken down into two molecules of pyruvic acid or pyruvate. Either one will be fine. Glucose, remember, is 6 carbons, 12 hydrogens, 6 oxygens, and we will see pyruvic acid is C3H4O3. We have 2 molecules of them because we will never be gaining or losing that carbon. So 6 carbon atoms here, 6 carbon atoms here. Looking at what happens in glycolysis is going to be what this process is called. I'll put this as glycolysis. Glycolysis starts with that glucose molecule. Now, I will say one thing in glycolysis, for it to start, we do have to make an investment of energy. You will have 2 ATP put into this reaction, and if 2 ATP are put in, remember that ATP will be broken down into 2 ADP and 2 PI, and that's your energy investment. Remember, to make money, usually you have to invest money. Make dollars, invest dollars first. Unfortunately, we're not going to give you some extra credit if you could tell me how to legally make a lot of money without spending a lot of money. All right, so we are initially adding ATP to glycolysis to prime or start that reaction. Luckily, at the end of glycolysis, you are going to be getting 4 ATP molecules out of glycolysis. And remember, if you have 4 ATP coming out of that reaction, you would have had to have the substrates 4 ADP and 4 PI. Those are just put together to produce those 4 ATP. So to look at 4 total produced minus 2 invested means you will have a net ATP production of 2. So you do get some ATP even though you had to invest a little bit. Even more importantly from glycolysis is we have harvested a couple of those hydrogens. Remember, those hydrogens also contain protons and electrons. In order for those protons and electrons to be harvested, we do need to have an electron carrier. We will be focusing on the electron carrier, NAD, and that NAD will be our empty truck going in to glycolysis. So here is that overview. Glucose will be initially phosphorylated with that phosphate, but then we will have enzymes that cut glucose apart, the lysis of glucose, into 2 molecules of pyruvic acid. During that process, the harvest of 2 hydrogen atoms will also take place. So enzymes are capable of harvesting a couple of hydrogen atoms. Remember, each hydrogen atom equals a proton and an electron, and electrons cannot run around the cell freely. They have to be carried. And so the hydrogen that is harvested from glucose will be loaded onto that electron carrier, which we call NAD. Now, NAD, in its loaded state, will be NADH. All right, so remember that is now reduced. Where are these NADH molecules going? They are going to those loaded trucks are going to take their harvest to the electron transport chain. All right, now let's get to the Krebs cycle. Remember our class, we are folding in the intermediate step. There is no entry step required for our class. So we are just going to have the Krebs cycle. And I consider the Krebs cycle as my chop shop. So that is my nickname for the Krebs cycle. So what happens in the Krebs cycle is these two molecules of pyruvic acid that were produced after glycolysis was lysed into two. Now the Krebs cycle is going to completely harvest every single hydrogen that is left. So every single hydrogen is going to be harvested off of that pyruvic acid molecule. So enzymes are going to chop that molecule completely apart, harvesting, chopping this molecule apart, chop. Now, all of the hydrogen has been completely harvested. There will be some addition of water, which will contribute some oxygen. However, we're not going to focus on that either. Do you see any more hydrogen left to harvest? No. That pyruvic acid has been completely broken down, and now you will see it into 6 molecules of carbon dioxide, because there were 6 starting out carbon atoms, and now those have been completely broken apart, harvested, and this is going to be excreted as waste. What else is happening in the Krebs cycle? Well, where did all these hydrogens go? Of course, they went to their electron carriers. There have to be many electron carriers available to collect that harvest to transport them to the energy market, which we call the electron transport chain. In fact, there will have to be 8 NAD molecules that are going into this reaction. So we'll have 8 NAD molecules that are going into the Krebs cycle. We will also have a couple of FAD molecules. Think of NAD and FAD as one's a Chevy, one's a Ford. They both are carriers. They just are slightly different brands, but they do still have the same function. They will also carry those atoms a little differently. 8 NAD will be loaded or reduced to become 8 NADH. And the FAD is able to carry 2 protons and 2 electrons, 2 FAD. But you can see that those molecules have been loaded up with their hydrogen or reduced. Do they have more potential energy or less? And where are they going? Well, they are also going to be ETC. So all of these energy trucks that are loaded up will be going to the energy market. CO2, that carbon backbone of glucose has been completely harvested, so CO2 is now going to be waste. Last thing that is occurring in the Krebs cycle is the slight release of some energy. So there is a couple of ATP that are produced, not much. Remember, whenever we have 2 ATP at the end of the arrow, you know you're going to have 2 ADP plus 2 PI at the beginning. There cannot be a product without substrates. All right, What are the substrates of the 6CO2? Substrates would be that starting glucose. And then if you really are good at accounting for all of these atoms, you do see the 6 carbons here and 6 carbons here, but we have 12 oxygens. We only started with 6 oxygens up here. This is where I said there is the addition of some water. Some of that water will contribute that oxygen that you see here, but we're not going to account for that. Trying to simplify it as much as possible because you already have enough to memorize in 1 week. All right, so there's the Krebs cycle. Now for the exciting part, which is going to be the electron transport chain. So now we'll have the number 3, electron transport chain. So this happens to be that chain of respiratory enzymes. They will be embedded in a membrane. I will expect you to be able to draw this electron transport chain. So here's your membrane. Remember, in bacteria, it's the cell membrane. In eukaryotes, it will be the mitochondrial membrane. The important things to remember here will be the names of the very first enzyme in this chain and the very last one. So the first enzyme is going to be called Nadh dehydrogenase. I'm trying to remember which of the colors I am going to use for this. How about I will go ahead and just use blue. So this first enzyme is going to be called NADH dehydrogenase. There, NADH dehydrogenase. There will be some intermediate enzymes. All of these are embedded in my membrane. The last enzyme at the end of the chain is going to be called Atp Synthase. You will be expected to memorize NADH dehydrogenase and ATP synthase. Remember that they are embedded in a membrane, mitochondrial in eukaryotes, cell membrane in prokaryotes. All right, we know these are enzymes, specific three-dimensional shaped protein with a specific substrate that they will bind to, and they will perform a particular function on that substrate. What is the substrate of NADH dehydrogenase, and what is its function? Hopefully, you're thinking, using the clues in the name, it looks like NADH may be the substrate of this enzyme, and you would be correct. So remember, from glycolysis and the Krebs cycle, we will actually have 10 NADH, and that NADH as a substrate is going to bind to that enzyme. What's the function of NADH dehydrogenase? Dehydrogenate means removing the hydrogen. So that hydrogen will be removed. Now NADH has been oxidized, and now this oxidized NAD, as the empty truck, can go back to Krebs and glycolysis for more protons and electrons. Remember that H will be those protons and electrons. So I'm just going to carry my NADH coming down from glycolysis in the Krebs cycle. Now, can you see that that NAD can go back to the Krebs cycle and back to glycolysis, so that we can cycle. Large molecule of glucose, here's my pyruvic acid. Remember the Krebs cycle has completely chopped apart that Krebs, completely chopped apart, pyruvic acid into little bits of individual carbon dioxide molecules. Every single hydrogen has been harvested, and remember those hydrogens will be loaded onto the electron carriers, the trucks, to go to the energy market. Do you want to be able to recycle those trucks? Especially if you were an apple farmer, you do not want to be buying a new truck for every harvest. So you will be recycling these tools. It is a coenzyme, it's not an enzyme, but it will be recycled like a tool. All right, so 10-NADH as the substrate, binding to NADH dehydrogenase. That enzyme takes off, removes the hydrogen, dehydrogenates it. Now it's unloaded or oxidized. It can return to the Krebs cycle and glycolysis to pick up more hydrogen. Now let's talk about what happens to this hydrogen at NADH dehydrogenase. There will be protons that are going to be kicked out of the cell. However, those electrons, remember, are very volatile. So electrons have to be very carefully transported. As they are being transported from one enzyme to another in this chain of respiratory enzymes, the enzyme that has the electrons is reduced, and when it gives it away to the next enzyme in the chain, now this one is reduced and this one is oxidized. This one is reduced as it gives it away to the next one in the chain. That next one is reduced, and now this one is oxidized. It's kind of like a person playing catch or sandbags that are being passed down. Whoever has the sandbag or the ball is reduced. They have more potential, versus when they give it away, now they are oxidized, less potential, but the next molecule has that extra potential or extra energy. Now, this is a series of oxidation and reduction reactions. So this one enzyme is reduced. As it gives the electrons away, it's oxidized, and now this one's reduced. So there's that redox reactions. As these electrons are moving through this chain of enzymes, there will be additional protons from the cytoplasm that will enter through those enzymes and be pumped out. Now, one thing I'm hoping that you're starting to see is there is the high concentration of protons on the outside of that membrane, and the electrons are in the inside of this membrane. It's setting up what looks almost like this. Have you ever seen something that has positives on one side and negatives on the other? That would be your battery. To charge that battery, you need to separate those charges. Can you see what NADH dehydrogenase and these other enzymes are doing? Separating those charges. As they establish that gradient of charge and chemicals, because protons are charged, they cannot just diffuse through the membrane. They cannot just go back and forth through that membrane. Any diagram you see, including our textbook, that shows those as traveling straight through the membrane is not correct. These charged ions will be traveling through a protein channel. The only protein channel that allows them to move into the cell happens to be the enzyme at the very end of the chain. As those protons move through this proton gate, they generate what we call the proton motive force. So protons moving through this enzyme create a force. That force energizes this enzyme, ATP synthase. And why does ATP synthase need energy to perform its main function, which is to synthesize ATP? So I will have ATP synthesized down here. Remember, if there is an end product, there have to be substrates up at the top. So we will have ADP plus PI as your substrates that are bound together. But like I've said before, this is an anabolic endergonic reaction that does require energy. Where is the energy coming from? This proton motive force with the difference in charge of protons and electrons, as well as a chemical gradient, just protons and electrons that are separate from each other. There will be 34 ATP that are produced by the electron transport chain in aerobic respiration for bacterial cells and for mitochondria as well. All right, we're almost done. Just like I said, these protons are going to be flowing through this enzyme ATP synthase. The electrons will come off the chain as well. Do we want those electrons running freely around our cells? By no means do we want that. So there does have to be that final electron acceptor. What's the final electron acceptor? Oxygen. So oxygen is input. So it is inhaled, and it will serve as the final electron acceptor. And see what happens when oxygen accepts 2 electrons and 2 protons. Hopefully you'll be able to see that those 2 protons and 2 electrons will become H2. The oxygen will be oxygen, and now we have metabolic water. Woo. All right. A couple of the questions in the study guide. One asked, what is the job of NADH dehydrogenase? I'm hoping you can explain the job of NADH dehydrogenase as binding to its substrate, NADH, and it will unload, dehydrogenate, and oxidize NADH. All of those are similar, so that NADH has that H removed. NAD is now unloaded in an oxidized form that is allowed to go back to the Krebs Cycle glycolysis to pick up more of the harvest. Meanwhile, let's put the harvest to use. Protons will be shuttled or pushed out of that enzyme to the outside. The electrons have to be carefully transported from enzyme to enzyme. While those electrons are being transported, those enzymes will also allow more protons to move outside, and they will actually force those protons outside of the membrane. Why do we want that large gradient? Because the larger the gradient, the larger the force of those protons as they want to go down their concentration gradient and charge gradient to establish equilibrium. They don't want to be more on the outside and less on the inside. They want to get to that equilibrium. That movement of those protons will energize that enzyme, which catalyzes this chemical reaction. These we do not running around the cell though, so there does have to be that final electron acceptor. What happens if oxygen is not present? This process cannot take place. So the only reason we are utilizing oxygen is to serve as that final electron acceptor. Do you see oxygen coming to play anywhere else up here? No. But if we do not have a particular ingredient in our recipe, does the final end product of our recipe get made? No. that is why oxygen is one of those vital signs that is measured in patients with those pulse oximeter monitors that are usually applied to the index finger, measuring oxygen saturation, which we are hoping is around 95% oxygen saturation. Why do we want a lot of oxygen? To produce a lot of ATP, because patients that are in a hospital do require a lot of ATP to heal and have all of those cellular processes take place. All right, hopefully you will be able to redraw that because he's pretty amazing, and you can watch the animations of the electron transport chain that I have linked in our